So welcome you back, my dear students. Uh, we are actually discussing about the RNA and its processing and the types of RNA. So it's a very big chapter, you know. So maybe we'll need another more 2-3 classes on that to complete the RNA processing. So during last 3-4 classes, we have learned about several things about the RNA, how RNA is processed, and what are the things, what are the event actually happens in the nucleus, in the cytoplasms, and how immature RNA is formed, then immature RNA to mature RNA is transferred using RNA ribonuclease and RNA polymerase enzymes. So these are the things. So here in the cytoplasm and in the nucleus, here you can see and how RNA processing in the nucleus or nucleolus, the primary transcript. Primary transcript, you know, we have already discussed in the last video. Primary transcript means the initial transcribed mRNA or RNA from DNA to RNA via transcription. Okay, so after transcription from DNA to RNA is from, so where exon and intron both the parts will be there. So this is nothing but the primary transcript. So primary transcript are the combination of exons and introns, means the coding and non-coding regions, and from where only coding, means the exons part will be separated using enzymes. And then once it is separated, so it will termed as a mature mRNA. Okay, so which will take part in the translation process for amino acid synthesis. So removal of nucleotides, here you can see from the primary transcript, removal of nucleotides, then addition of nucleotides to the 5' or 3' ends. So you can remember the mRNA structure. mRNA structure means 5' is the header portion, is the tailor portion, 3' is the tailor portion. So after header portion, there will be a initiation codon, then protein amino acid synthesis codon, then a termination codon, then 3' means the trailer position. This is the overall structure of mRNA. Okay. For your quick reference, we can show you here. So here you can see it's the typical mRNA structure. Okay, UTR, it is called UTR, 5' UTR. UTR means untranslated leader sequence. It can lead. And here is the translation start, initiation codon. In between the protein sequence, protein amino acid synthesis codon, here is the translation stop codon. And 3' untranslated trailer region, 3' UTR. Okay, so this is the thing. Then modification of certain nucleotides. So modification, why it is required? Because if there is any damage portion, damage point mutation if occurred, then one triplet codon also will be affected, so amino acid synthesis will be hampered. So then ultimately we will have the mature RNA. Okay, so mature RNA will take part in the translation process. So what is the things actually happens in a word? You can see removal of nucleotides by both endonucleases and exonucleases. So nucleases actually further divided into two types. One is the endo and another is the exo. Endonucleases means it cut inside the sequence. Okay, endo, and exo means it cut at the either it at 5' or 3' at the end portion. Okay, means outside the sequence. So endonuclease to cut the specific site within a precursor RNA, and exo is the trim of the ends of the precursor RNA. Here it is already defined, and this is the removal part. Okay, and what about the addition part? Addition part means then once it is removed, then the addition of nucleotides to 5' or 3' ends of the primary transcript or their cleavage product. So that means at the 5' end or 3' end of mRNA, the primary transcript will be added. Add a cap and a poly A tail to pre-mRNA. So poly A means a lot of adenine base. Adenine bases will be there. It will act like a cap. Add a cap and a poly A tail to pre-mRNA. So this is not the mRNA, it's the pre-mRNA. So at the 5 prime or 3 prime end. Then what it actually happened? Then modification. Modification means of certain nucleotides on either the base or a sugar moiety. Means is if anything is damaged portion is there, then suppose here you can see is a methyl group, methyl mismatch DNA repair, methyl group or ribose in mRNA and rRNA may be added here. Okay, so generally methyl group is actually added because methylation process will be started for translation initiation. Okay, so in the mRNA processing, what are the things actually is the overall conclusion? Genetic information is transferred from gene to the proteins. They encode via a messenger RNA intermediate. Okay, so mRNA is actually the thing, whole thing, the genetic information it carries. And several features is the processing of mRNA, hnRNA, snRNA particles, 5 prime capping. Okay, 5 prime capping. Capping means the cap like the poly A tail means the adenine, several adenine will be added at the 5 prime end. This is called capping, like a cap. And 3 prime cleavage is the cut 3 prime. Polyadylation means the poly a tail addition of poly a tail. Splicing is also important. Splicing means the cut portion is the unwanted bases will be cut. Pre-mRNA methylation. Pre-mRNA methylation means the addition of methyl group and rRNA group, mRNA group. Okay, so here is a very important slide, characteristics of the 5 RNA polymerase of eukaryotes. Okay, this is a very important slide. You can see the enzyme RNA polymerase 1, 2, 3, 4, and 5. See here. So the location of RNA polymerase is the nucleus for all. Only RNA polymerase 5, 4, and 5 can be visible or can be found in plant system. Okay, and RNA polymerase 1, 2, 3 can be found in all the system. Where it is, why it is mentioned here? Only it is found in plant system. That's why. And their product, see here, this is a very important slide. Again, I repeat you. So RNA polymerase 1, its function is ribosomal RNAs excluding 5S rRNA is the product. Okay, so RNA polymerase actually it act as a cleavage or splicing enzyme, means the primary transcript when will be transferred into mature transcript, so polymerase will help there, and the product will be developed like the ribosomal RNAs excluding 5S rRNA. Nuclear pre-mRNAs or RNA pol 2 is also short, in short we call is RNA pol. Okay, RNA pol 2 is the nuclear pre-mRNA. Pol 3 is the tRNA, 5S rRNA, and other small nuclear RNAs. 4 is the small interfering RNAs, siRNA is the very important. And 5 is the some siRNA plus non-coding means the antisense transcript of siRNA target genes. Okay, so this is the overall RNA pol product. So now we can start about the mRNA processing in prokaryotes. Okay, mRNA processing in eukaryotes is already done. Now we are talking about the prokaryotes. So there is a little or no processing of mRNA transcript in prokaryotes. In fact, ribosomes can assemble proteins before mRNA molecule have not yet been completely synthesized. Okay, so this is not so critical for prokaryotes mRNA processing, but prokaryotic mRNA is degraded rapidly from the 5 prime end therefore only be translated for a limited amount of time. So this is a very important feature for prokaryote. In case of eukaryotes, this is not rapidly degraded. But in case of prokaryote, you can see the mRNA is degraded rapidly for 5' end. So therefore, only translated for a limited amount of time. So for a very short while. Okay. So here in the picture you can see, so eukaryotic mRNA transcript the process. Here you can see is the nucleus is total. This is the nucleus, this is the cytoplasm if we talk about a eukaryotic cell. So in the nucleus there is the DNA, and DNA after transcription will process the mRNA or RNA. This is called the primary transcript. Okay, so this RNA is called the primary transcript, means there are exons, introns, all the things are there. Then RNA will be processed. RNA processing means the primary transcript will be converted into mature mRNA. Mature mRNA means all the non-coding part will be removed, damaged portion will be removed, methyl group will be added. So it will be completely mature RNA so that it can take part in the translation process. So RNA processing, then mRNA will be produced. So mRNA will be produced once, then poly. See here, poly A tail means the cap, poly A cap, so that 3' will not be damaged. So poly A cap will be added here inside the nucleus. Then it will export from the nuclear pore. You can see the nuclear pore. This is the nuclear membrane, and see there are the nuclear pore. So it will be exported into the cytoplasm via nuclear pore, and then will attach and bind to the ribosomes, the small subunit and large subunit ribosomes. Then tRNA will come here, then mRNA along with tRNA and T codon amino acid synthesis will be started. So this is the overall process. Okay. So I think up to this one we can stop here. We can start in the next class from here onwards. Okay. Thank you for watching.